Bismillahirrahmanirrahim. Assalamu alaikum students. I am back with the part 2 of the chapter inheritance. Today we are going to discuss about the mutations, variations and selection and you can say the diseases which are related to the mutations. So starting with the variation, there are two types of variation. Number one is discontinuous and the continuous variation. If there is a clear-cut phenotype with no intermediate between, like my blood group is A, or it can be B, it can be AB, or it can be O. So such type of variation is known as the discontinuous variation. It is having no intermediate in between. I don't have the blood group A dot B like B dot O. I don't have such type of the blood groups. My blood group either can be A, B, AB or O. I don't have the C, D, E, F, G such type of blood groups. Fine. So such type of the variation is known as the discontinuous variation, and these variations are found in the traits or the features which we are going to inherit from our parents. And these particularly are being expressed in the form of phenotypes. We are not talking about the genotype. They are only expressed in the phenotypes. Some people are having the ability to roll their tongue. Some don't have. There is no intermediate between that I am going to partially roll the tongue. No, there is no such kind of scene. Either my earlobe is attached to the chin or that is free. I am having the widow's peak or don't have. Such type of things are known as the discontinuous variation. Some people are having 6 fingers. Either they are having 5 or the 6 fingers. Some people are having joined fingers, joined limbs are over there. So they are having or not having. So there is no intermediate case between these conditions. So such type of things are known as the discontinuous variation. While talking about the continuous variation, which are with any trait, with any phenotype, which is having multiple choices, which is having many phenotypes, a range of phenotype, or which is having many intermediates in between. Such type of variation are known as continuous variations. Like human height, everyone can have different height. You might have heard there are dwarfs. Some are having height of 3.12, 3.45 like that, 4.12, 3.45, such on and so on. So there is a variety of height. It is different in different population. Even in a home, in your family, you all will be having different heights. Talking about the weight is also example of continuous variation. Everyone in your home will be having a different weight. Like if my weight is 71, my daughter is having a weight of 42, my son is having weight of 52, my husband is having weight of 79 point something like that. So the weight is also different among the population, among within the family even. So that is different. Intelligence is also different in every family member. So definitely if we are having different intelligences level at our home, so it is also different in the population as well. These particular variations, which are known as the continuous variation, they are affected by the genes, plus they are affected by the environment. Along with that, there is a thing that these are not only controlled by a single gene, they may be controlled by multiple genes. This is the example of your continuous and discontinuous variation. There is no intermediate in between. That is the discontinuous variation, and height that is different in the different populations. So that is the continuous variation. Many phenotypes can be observed, while in this case only the 4 phenotypes are being observed, while here you people can see different phenotypes are available. So that is the difference between the discontinuous and continuous variation. I have told you that not only the human they are having the discontinuous variation, but in within the organism like plants and animals, they are also showing some discontinuous and continuous variations. This is one important point. The discontinuous variation may be controlled by one or few genes, while many genes are involved in controlling the continuous variation, and they are going to show the additive effects. And continuous variation is affected by the genes plus environment as well. Like if I am relating to you people, giving the example, very best example, if you people are getting the opportunity to do the physical exercise from your childhood, or even you people are doing it right now, then you people will be having higher length or more length as compared to the other who are not physically active. If you people are having sufficient amount of food to eat, then your weight will be more as compared to the people who are starving. So ultimately your length, body height, body weight is going to be change according to your environment. जितना ज्यादा खाएंगे वेट उतना ज्यादा हो जाएगा, जितने ज्यादा फिजिकल फिटनेस या एक्सरसाइज करेंगे हाइट उतनी इंक्रीज होगी. So they are related not only with the genes but with the environment as well. So you can see the diagram. The skin color is a variety in which multiple colors you can be seen over here. Height, skin color, weight. These are the three pet example of the continuous variation. While blood group, ear lobed, you people are having lobed ear or not, widow's peak or not, tongue rolling, all these features are discontinuous variation. In the plants, either they are round, either they are wrinkled, either they are yellow or the green, such kind of features, yellow flower, purple flowers, they are the example of discontinuous variation. And continuous variation, they are affected by the environment. Okay, let's do a question. For example, the height of 500 pea plants of the same age were measured to the nearest 20 centimeter. The results are shown. Variation in height of these pea plants show which kind of variation, continuous, discontinuous, both or neither. Okay, अब आप देखें, just see height is the example of continuous variation, which is being discontinued in the range of 121 to 140. So it means that the 42 or the C option is right. Height is example of continuous variation, which is being discontinued in this range. ठीक है जी, तो that's why that is considered as the continuous and discontinuous variation both. This is the another question related to the mass, which is also the example of continuous variation. Genes do not affect the mass of tomatoes. No, genes are going to affect. Genes affect the mass, not only of the tomatoes, but everyone. Type 1 tomatoes show continuous variation. Yes, that is the example. Mass is the example of continuous variation. Type 2 tomatoes are sometimes smaller than type 1 tomatoes. No, that is not obvious from the picture. Type 2 are more in height. That is not right. Type 2 tomatoes show discontinuous variation. No, mass is the example of continuous variation. So option B is right over here. Now coming towards the mutation, you all are very much well familiar that genes, they are being transferred from parent to their offsprings. And whenever talking about the meiosis and the mitosis, meiosis is happening inside the gamete. Cell is going to divide, and that is going to replicate its copies, which are transferred to the offspring. And to transfer it in the accurate number, accurate copy, the genes or the chromosome should be divided properly. If any error is going to occur during that replication, either in meiosis or mitosis, then definitely some results will be very much different. And if these are going to happen in the gene and the chromosome mutation, that is known as the mutation. If that is going to happen inside the gene or the chromosome, it cannot be changed, it cannot be reverted, it cannot be cured. This type of condition is known as the mutation. Mutation is a sudden random change, either in the gene or in the chromosome. And that is also the responsible for diversity in the species. Mutation is responsible for diversity in the species, but mutation can be responsible for some diseases such as cancer, such as sickle cell anemia, such as Down syndrome. Okay, so there are two types of mutation which will be involving the genes and which will be involving the chromosomes. On the basis of type of cell which is which in which the mutation is happening, either somatic or your body cell, they are going to face the mutation, or the gametes, they are going to face the mutation. If it is your somatic cell, then what will happen, this condition is known as the cancer. आपके बॉडी के सेल अनकंट्रोलेबली प्रोलिफरेट कर रहे हैं, दैट इज अ म्यूटेशन. अनकंट्रोलेबली डिवाइड हो रहे हैं, दैट इज अ म्यूटेशन, एंड दैट इज नोन एज द cancer. If we are talking about the body or the somatic cell. But if that is the case for the gametes, then ultimately we are talking about the genes, we are talking about the chromosomes. So that can be more, you can say, dangerous condition, because cancer is there are chances of the cancer to be cured, but once the mutation is happening in the genes, ultimately that cannot be cured, that cannot be repaired. The mutagens from examination point of view, these are very much important. Remember, UV radiation, formaldehyde and the tar in cigarette smokes, beta gamma radiations, all they are mutagens. LSD, that is lysergic acid diethylamide, that is also a mutagen. So please remember the names of mutagen. They are going to cause a sudden random change either in the gene or in the chromosome or in the somatic cells. ठीक है जी. So now coming to word, that is a very much comprehensive slide which is showing you people if the mutation is happening inside the genes. Then two example in your syllabus are the albinism and the sickle cell anemia. While if it is happening in the chromosomes, then the Down syndrome is happening. You can have a screenshot of it. So I will be telling you these things in detail. Okay. So somatic mutations are related with the body mutation, and they are responsible for the cancer. They are not going to be passed from the parent to the offspring. जिस person को है सिर्फ उसी को है ये disease. Offspring में जाने का chance नहीं है. This is condition known as the albinism, and that is the example of recessive gene mutation. इस condition को show होने के लिए recessive genotype होना जरूरी है. ठीक है. सो अगर पर्सन इज हेट्रोजाइगस तो अल्टीमेटली उसके अंदर एल्बिनिज्म नहीं होगा. ठीक है. इफ आई एम गोइंग टू टेल यू पीपल, अगर इफ यू पीपल हैव सीन द पार्ट 1 ऑफ द वीडियो, यू माइट रिमेंबर कि मैंने आपको पुनेट स्केयर के थ्रू बताया था कि होमोजाइगस और हेट्रोजाइगस क्या होते हैं. दिस कंडीशन इज नोन एज द हेट्रोजाइगस डोमिनेंट, और ये आपके पास हो जाएगा होमोजाइगस रिसेसिव. तो ऐसी कंडीशन अगर इसमें है, then that will be the albinism. अगर ऐसी condition है, then there is not the albinism, but that person is the carrier. अब if that is going to be crossed with the same heterozygous, तो आपको पता है कि हमारे पास यहाँ पे एंड पे बीबी आने का चांस है. तो such type of parents will be having 3:1 होगा उनमें, कि बेबी में 4 में से हमारे पास 1 का चांस है कि that is having the albinism. Okay, next is that individuals who are homozygous for the albinism allele are known as the albinos, and that is characterized by the absence of the melanin pigment in the skin, in the hair, and iris is not having any color. Such persons are very much sensitive to the light. They can have the sunburns very much quickly. Their eyes become or they appear red because they don't have any pigment in the choroid layer of the eye. They don't have any pigment in the iris. So the blood vessels, they are reflecting their color, which is red. Okay. That second condition is gene mutation is sickle cell anemia, in which particularly the blood cells, red blood cells, they are having a crescent shape. Usually that is considered S shape, but that is a crescent shape. And that is because of a change in the sequence of bases or the nucleotide. That is particularly because of change in the sequence of bases or the nucleotide in the DNA. Normally, person is having HbA, but in this condition of the sickle cell anemia, person will be having HbS, that is the sickle-shaped hemoglobin. You are very much well familiar that hemoglobin, they are responsible for transportation of the oxygen because oxyhemoglobin is formed. But in this case, whenever the hemoglobin is not normal, that is sickle shaped, then what will happen? Its capability to transport oxygen will get reduced. So such person will be having the breathing disorder. They will be having lack of oxygen inside their body, and definitely that oxygen के ऊपर जो process depend कर रहा है, that is the respiration, ultimately that will be affected. So normally what did happen कि हमारे पास that is a normal base sequence. You might have seen the part 1 in which I told you GAG, that is the guanine, adenine and guanine. These are the bases shown to you people. Only a single change, that at the place of A there is T. गलती से GAG की जगह GTG आ गया है. So these nucleic base nitrogenous bases are responsible for coding one amino acid. तो जो amino acid normal बनना था उसकी जगह पे वेलिन म्यूटेंट आपके पास अमाइनो एसिड बन गया है. एंड अल्टीमेटली एक गलत अमाइनो एसिड किसी भी चेन में फिट होगा तो एक गलत प्रोटीन ही बनेगी, एंड दैट रॉन्ग प्रोटीन इन दिस केस इज द सिकल्ड शेप्ड हीमोग्लोबिन. Hemoglobin is a protein. ठीक है. So A is going to replace is being replaced by the T, and the triplet code is going to be changed for the amino acid, and that is only the changing in the gene mutation. Do you people know that the sickle cell disease is a very much fatal condition? सिकल सेल डिजीज क्या होती है? जिसमें आपके पास जो फादर और मदर दोनों बोथ वर द कैरियर ऑफ द सिकल सेल trait, and they are going to inherit the disease to their offspring. Such people will be having low concentrations of oxygen in the blood, and ultimately oxygen carrying capacity of the red blood cell because of the sickle shaped hemoglobin is reduced, and they are becoming more fragile. Such persons can they are having very much short lifespan. सो सिकल सेल एनीमिया के लिए होमोजाइगस रिसेसिव कंडीशन होती है. पर्सन हैज टू बी होमोजाइगस, दैट इज अ रिसेसिव कंडीशन. You people can see the sickle shaped RBCs which are not normal. That is a carrier person. इसमें RBCs normal भी है, but some are the sickle shaped. So such person, they are the, if that is the same, this is the condition, such person are the carrier. They are comparatively uh, in less danger as compared to the person who is having sickle cell disease. This is the sickle cell trait, that is the sickle cell trait, जिसमें एक पर्सन में जो जीन्स उसने फादर और मदर दोनों से लेनी है, उसमें एक में है और एक में इसको नहीं मिला, ट्रांसफर नहीं हुआ. That is heterozygous condition. In this condition, RBCs are shown you people the heterozygous form. Some are sickle cell, some are normal. Do you people know that this sickle cell anemia is the deficiency of blood because of the lack of the hemoglobin? Such person who are having the sickle cell trait, which are the heterozygous. Disease condition may they will be the homozygous. In the heterozygous condition, they are having a chance of better survival if the person gets the malaria. अब मलेरिया अगर किसी पर्सन को है एंड दैट इज अ हेट्रोजाइगस, उसमें हाफ आरबीसीज नॉर्मल है, हाफ सिकल शेप्ड है, सो दे विल बी हैविंग अ बेटर चांस ऑफ सर्वाइवल ऑफ द मलेरिया, बिकॉज मलेरियल जो पैरासाइट होता है प्लाज्मोडियम, दैट इज गोइंग टू multiply in the human RBCs. So whenever the RBCs are not the क्या कहते हैं उसको, आपके पास RBCs नॉर्मल नहीं है, कुछ सिकल शेप्ड हैं, तो उस पर्सन में person with the malaria that can survive. But यहाँ पर अब ये आपको एक डायग्राम नजर आ रही है, इसमें नॉर्मल आरबीसीज एंड दैट इज अ पर्सन विद द कैरियर दैट इज विद द सिकल सेल डिजीज. In this condition, I am going to compare the sickle shaped person with the disease with the normal one. ऐसी डिजीज कंडीशन में पर्सन का सर्वाइवल बहुत शॉर्ट होता है, उसका लाइफ स्पैन शॉर्ट है, बिकॉज आरबीसीज आर नॉट नॉर्मल, दे आर नॉट एबल टू ट्रांसपोर्ट ऑक्सीजन प्रॉपरली, तो उसका लाइफ स्पैन अल्टीमेटली कम होना ही होना है. Now talking about this person, the which is having the heterozygous condition, कुछ उसमें आरबीसीज नॉर्मल है, कुछ सिकल शेप्ड है. दैट पर्सन इज कंपैरेटिवली बेटर, उसका सर्वाइवल चांस है एज कंपेयर टू द सिकल सेल डिजीज पर्सन, बिकॉज उसमें कुछ आरबीसीज नॉर्मल ऑक्सीजन ट्रांसपोर्ट कर सकते हैं, बट इसका लाइफ स्पैन इस नॉर्मल वाले से कम होगा. ठीक है. if these two are going to do the exercise, ये बहुत जल्दी ए जो है हेट्रोजाइगस दैट विल गेट एग्जॉस्ट क्विकली, थक जाएगा, बिकॉज ऑक्सीजन सप्लाई में कम हो जाएगी. ठीक है. बट वाइल टॉकिंग अबाउट द मलेरिया, this person with the homozygous recessive is having a better chance of survival from the malarial parasite as compared to these two. बिकॉज ऐसी कंडीशन में हमारे पास पर्सन में वो नॉर्मल आरबीसी ही नहीं है जिसमें जाकर प्लाज्मोडियम जो पैरासाइट है, प्रोटोजोन है मलेरिया का, दैट इज गोइंग टू लिव, मल्टीप्लाई एंड सर्वाइव, वो कर ही नहीं पाएगा. ठीक है. इस कंडीशन में वो सर्वाइव कर लेगा क्योंकि इस शेप में हमारे पास पर्सन के पास नॉर्मल आरबीसीज नहीं है, ना प्लाज्मोडियम को आरबीसीज नॉर्मल मिलेंगे, ना वो सर्वाइव करेगा. ठीक है. आरबीसी शिकल शेप है, दे आर नॉट सपोर्टिव फॉर द मलेरियल प्लाज्मोडियम, सो पर्सन विल बी सर्वाइविंग फ्रॉम द मलेरिया, बट दैट कैन डाई बिकॉज ऑफ द लैक ऑफ ऑक्सीजन. सिकल सेल डिजीज होने का एक डिसएडवांटेज ये होता है, पर्सन की आर्टरीज और वेन्स के अंदर वो जाके स्मॉल आर्टरीज में जाके ब्लॉकेज कॉज कर सकते हैं. अल्टीमेटली first they are not going to transport oxygen, and on the other hand they are going to block the blood vessels, so they are causing a disease condition to a person. यहाँ पे आप देखें, that is the comparison between the carriers. ये हमारे पास carrier हैं with the sickle cell trait. They are having the offspring, 4 offspring, out of which one will be the diseased person, other 2 will be the carrier, and 1 will be the normal. अब इस condition में this will be the healthy person. These 2 are having the disease, but they are the carrier. They don't have the proper disease. Their lifespan is better as compared to this affected HBSS. Fine. And this person is having a better survival chance in the malarial condition, because malarial parasite plasmodium is going to live in the RBCs, and RBCs, whenever they are not the normal, they are not going to provide the suitable environment for the replication and growth of the plasmodium. So plasmodium will die itself because of the lack of normal RBC that will not be able to grow into the sickle shaped RBCs. So ultimately that plasmodium will get die. Okay. So I told you that heterozygous, they are having a survival advantage in case of the malaria, but the people who are homozygous recessive with the disease, they are having very much short lifespan. Lifespan of normal RBC is about 120 days, while in case of the sickle shape, they are having very much less lifespan. So such people can not survive for a longer period. They are having a short survival time. So that is Punnett scare shown to you people. These 2 are the carrier. These are the parents. And see, 1 is normal, 2 are the carrier, and 1 will be the diseased person. This is known as the sickle cell disease. AS is known as the sickle cell trait. They are the carriers. They are not neither normal, neither diseased. Okay. Now talking about the chromosomal mutation, the thing which is in your syllabus, the disease that is the Down syndrome. In this case, the person is having 47 chromosome in their bodies, and you people should know, you should recall that the person in which the number of chromosome are going to be changed, either that is going to 45 or 47, they will be infertile, they cannot reproduce, because during the cell division of the gametes, meiosis, main number of chromosome cannot divide equally, so such persons will be infertile. And in this case, there is an extra copy of chromosome in the form of triplet at the chromosome place number 21. ये जो क्रोमोसोम 21 है, वहां पर एक एक्स्ट्रा कॉपी इनके पास आ जाती है, 2 की बजाय 3 क्रोमोसोम्स हो जाते हैं. Can you people identify the gender of this person? Mention that in the comment box. Okay. During meiosis, either in the meiosis 1 or the meiosis 2, the disjunctioning के दौरान में हमारे पास क्या होता है कि एक्स्ट्रा कॉपी कहाँ से किस तरह से आती है. whenever these chromosome are going to divide, this is the normal division, but that is abnormal. This is not normal. This division is normal, but this division is not normal. ठीक है. सो इनमें से कोई भी क्रोमोसोम अगर आप रेंडमली मिला देंगे, दिस इज गोइंग टू फ्यूज विद दिस, तो अल्टीमेटली यहाँ पे 47 बन जाएंगे. इसको आप इसके साथ मिला देंगे तो यहाँ पे 45 क्रोमोसोम हो जाएंगे. ठीक है. सो दैट इज हाउ कि हमारे पास ड्यूरिंग meiosis 1 and meiosis 2, how the chromosome, they are not going to be separated, which is responsible for the extra copy from the parent in the offspring. These persons usually are having particular facial features, and they are have suffering with the mental and physical disabilities. Okay, here is a question. How many chromosomes are there in a zygote which develops into baby with Down syndrome? Mention your answer in the comment box. अगर अभी तक आपने मेरा चैनल सब्सक्राइब नहीं किया, प्लीज सब्सक्राइब इट एंड प्रेस द बेल आइकन सो दैट यू कैन गेट द नोटिफिकेशन ऑफ माय न्यू वीडियोस. ओके, सो म्यूटेजेंस आपको बोला था याद कर लेंगे. सो म्यूटेजेंस आर ओवर हियर, यू कैन लर्न दीज. प्लीज रिमेंबर देयर नेम्स. यूवी रेडिएशन, अल्फा बीटा गामा रेडिएशन, आपके पास स्टार है, एलएसडी है, फॉर्मेल्डिहाइड, ऑल दीज आर द म्यूटेजेंस. Now, talking about the mutation and the selection, mutation is responsible for the biodiversity, diversity among the species. Some mutations are beneficial and they are going to help the predator or the prey to camouflage for their survival. If the predator is going to be camouflaged with the environment, that will not be obvious, or that will not be visible to the prey, so it can attack on the prey and can have its food. While in case of the prey, if that is camouflaged with the environment, so ultimately that will not be observed by the predator, it can survive and continue its own kind, so it's ultimately that is going to reproduce more quickly. That is a Punnett scale shown to you people with the 4:0, 1:1, and 3:1. Have a screenshot of it. Now coming toward the selection. Nature is going to select the varieties which are more competitive, more resistant to diseases, which are better adaptive to the changes in the environment. Means survival of the fittest होगा. कहीं पे भी क्वेश्चन आ जाता है, आपको ओवर क्राउडिंग नजर आ रही है, तो अल्टीमेटली क्या होता है कि पीपल आर गोइंग टू, और ऑर्गेनिज्म आर गोइंग टू फाइट विद ईच अदर फॉर द फूड, फॉर द शेल्टर, फॉर द mating. So they are going to do the fight for food, basically for the nutrients, for the space they are going to fight. So the organism which is the fittest to the environment, that is going to survive, and that is going to reproduce and carrying its own kind further for the future. So that is known as the natural selection. Mutation is providing the new genes and alleles for the natural selection to operate on. The new breed of the organism may be better adaptive to their new environment. So that is the selection. You can have a screenshot of it. While the evolution and the natural selection, they are also having a relationship. That is a comparison of the two, artificial or the natural selection. Natural selection is usually a slow process that is done by the nature, depending upon the fittest organism who is going to compete for the food and the environment. While artificial selection is a quick process which is done by the human, it takes less time. What are the factors would have to exist for a species to be able to change? These are the factors. There is nothing to be explained. You can have a screenshot. You have to learn these points. And that is about the artificial selection. किसी भी हमने चीज को आर्टिफिशियली सिलेक्ट करना होता है सबसे पहले ब्रीडर इज गोइंग टू सिलेक्ट द प्लांट दैट इज प्रोड्यूसिंग द सीड्स विद द high oil content. I am talking about the I want to get high oil content. The yield of oil should be more. So comparatively better, see the seeds with the better or the higher oil content are being selected. Good seeds, which are healthier one, they are going to be germinate. They are allowed to germinate. After germination, they are allowed to self-fertilized. And then the generations are being produced, which are further mated with each other. And after many generations of pure breeds, plants with the desired quality can be obtained. Please note down the slide. You can have a screenshot of it. और अगर कोई भी आपको artificial selection के लिए कोई भी feature आ जाता है, then you can write these 5 steps. आपने बस उसका जो जिस चीज के लिए आप उसको करवाना चाह रहे हैं reproduce, that is going to be replaced over here with the high oil content. Coming toward the hybridization, that is the cross-breeding of different varieties of plants or the animals to produce the new or the better variety. Hybridization is the example of the artificial selection. Note down the slides because there is nothing to be explained. आपने simple इसको read out करना है और इसको आपको याद करना है. इसमें कोई explanation वाला point नहीं है. That is a hybrid. That is a hybrid. Okay. Here is I'm going to tell you people the example of selective or artificial breeding. Jersey cows which are going to produce a good amount of milk, but they are unable to survive in the warm climate. They are being crossed with the Brahman cattle which are able to survive in the warm climate. So after multiple crosses, you people can get a cow with the good milk production with the higher survival chances in the warm climate. Here I just want to explain the point. The inbreeding can result in the accumulation of recessive alleles in the population, which can be very much dangerous if this recessive alleles is related with the diseased condition. ठीक है. So in breeding आप organism की self breeding करवाते जाएंगे तो ultimately recessive allele accumulate हो जाएंगे, and if the two recessive allele are going to be expressed in the organism, that can be a disease condition as well. Here is a question: which statement describes an example of artificial selection? It has been found that some strains of bacteria produce antibiotic that is not controlled by human, that is natural selection. It is common practice to mate bull with the cow that produce the most of the milk. Yes, that is. It is possible to control caterpillar on food crop by releasing small wasps which lay their eggs in caterpillar and kill them. That is the biological control. That is artificial selection, that is also, but that is biological control. We are going to control the amount, we are not going to produce something. ठीक है, artificially कर रहे हैं, by human हो रहा है, but that is not the selection. We are going to cancel out the things from the environment. Mosquitoes have developed strains, no, that is mutation. ठीक है जी, answer is B. Now coming toward the most important part, production of human growth hormone in genetically modified bacteria. You can replace the word growth hormone with that of the insulin. ठीक है जी. So these are the basic steps. ये मैंने लिखा आपको इंसुलिन के दिए हैं सारे के सारे steps, ताकि आप ग्रोथ हार्मोन अगर लेना है तो ग्रोथ हार्मोन कैन बी टेकन फ्रॉम द पिट्यूटरी ग्लैंड. ठीक है, पिट्यूटरी ग्लैंड से लेंगे एंड दैट इज बीइंग कट विद द रिस्ट्रिक्शन एंजाइम. आपने बैक्टीरियल प्लाज्मिड यूज करना है, डीएनए लाइगेज से बाइंड करना है, दोनों को रिकॉम्बिनेंट प्लाज्मिड बना देना है, इकोलाई में इंसर्ट करवाना है और फर्मेंटर में उसकी प्रोडक्शन करवा लेनी है. बेसिक जो आपने स्टार्टर जो करना है, इसमें आपको रिस्ट्रिक्शन एंजाइम इज अ कीवर्ड विच इज गोइंग टू मेक अप द स्टिकी एंड्स इन द प्लाज्मिड एंड द जीन फ्रॉम द पेंक्रियाज और फ्रॉम द पिट्यूटरी. Pituitary से ले रहे हैं, you people are going to take the growth hormone, and from the pancreas you people are taking the insulin. ठीक है, अब आपके पास प्लाज्मिड जिसके अंदर आपने इसको रिक्वायर्ड जीन को आइदर दैट इज फ्रॉम द जीन फ्रॉम द पिट्यूटरी ग्लैंड फॉर द ग्रोथ हार्मोन और फ्रॉम द पेंक्रियाज दैट इज द जीन फॉर इंसुलिन ले रहे हैं, that is going to be inserted into the plasmid with the help of DNA ligase. That plasmid will become the recombinant plasmid, which will be inserted into the E. coli, which will become the recombinant genetically modified organism. GMO बन जाएगा. ओके, यहाँ पर ग्रोथ हार्मोन ले रही हूँ, आपको बता रही हूँ, एग्जाम्पल दे रही हूँ. ग्रोथ हार्मोन लिया पिट्यूटरी ग्लैंड के सेल से, इसको आपने कट किया विद द रिस्ट्रिक्शन एंजाइम एंड आर गोइंग टू बिकम वेरी मच स्टिकी. एंड दीज स्टिकी एंड्स विल बी कॉम्प्लीमेंट्री विद दैट ऑफ द प्लाज्मिड, जिसको मैंने दोबारा से कट कर देना है रिस्ट्रिक्शन एंजाइम से. After that, I am going to bind it with the help of the DNA ligase. And then DNA ligase is going to make it place it's on its proper position where it is required. That has become the recombinant plasmid. Now I am going to insert that recombinant plasmid into the E. coli with the help of heat shock, electric shock, or the chemical treatment. Then that is being cultured into the fermenter. Whenever they are going to multiply, the required growth hormone or the insulin can be collected. That is a pictorial diagram for that. Okay, here is a question. Note down the question and give me the answer in the comment section. Another question for you. Answer is required to be written in the comment section. Note down the question and mention the answer in comment section. Okay, so here are some sources of genetic variation. Thank you very much. Hope you have liked my video. Please give me thumbs up and do subscribe my channel. Press the bell icon. Please share my channel with your friends as well. Remember, students, in this today's lecture that was for you people to learn the content, there was very much less points to be explained. So that's why the section in which the explanation or the concept is required, that is my favorite part. But the part in which the learning of the student side is involved, so I do not I cannot make it explain very well. So please आपने जो चीजें आपके याद करने वाली हैं आप उनको याद करने अच्छे तरीके से, because my grip, my command is on the explanation part with the logic. I hope you liked the video. Please share it. Thank you so much. Thanks for watching. Allah Hafiz.